Okay. When I started working here in the program, I saw it was very common things like today's event. You have a professor, you have something scheduled, and suddenly they go, they can make it. And many of our adjunct professors are PAs that are working full time. You know, when you work in healthcare, you know when you start, but you don't know what time you leave. Maybe you have a surgery that starts at noon, and it's going to be a couple of hours. You never know. Surgery can get complications, and you may stay there for many hours. Okay, so at the beginning I didn't understand why this happened. This should be more organized. I always I'm not OCD, I'm everything except OCD, but for time I have to be there at 8 and 30 minutes earlier. And I didn't understand, but then I understood. This is what really prepares them for what they are going to be doing the rest of their life. It's not that this is good, okay, but you get that flexibility, you understand that change is not always for bad, it sometimes has good things. Well, today we are going to start with this topic. We're like continuing from what we were talking about, the cells, okay, to enter into the cell membrane, the cytoskeleton. Okay, we were talking the other day about the functions of some organelles and how these functions relate to the synthesis of proteins, okay, from DNA, RNA to different proteins. And we are going to continue talking about the cells, mostly the cell membrane, cytoskeleton, and then we are going to focus on the transport mechanisms across cell membranes and also through epithelial membranes. And here we have something that I want you to once in a while read. This is at the end of the previous PowerPoint. Okay, and now we have it here. This is what we call the core principles in physiology. Okay, there are many, as you see there. Okay, I put an arrow on the ones that we are really going to be exploring the most. Okay, these are topics that are called core principles because they apply to every system. Okay, we divide, for example, the anatomy. You study anatomy in different ways, or using the different systems, or from head to toe, the regional anatomy. Or you can study the physiology in different ways. Typically we divide it in systems, but there are topics that will appear in every system. Okay, and why it's important to understand this? Because let's say we have an exam and you study homeostasis, you study the cell membrane, the mechanisms of transport, absorption, passive diffusion or active transport, this and that. And a mistake that some students make is, okay, the first exam is until the PowerPoint 5. Okay, I got an A. Now I can forget that and start with the new material. That is not true. Okay, because all these topics that you see here will appear over and over. And when we move into the fall semester pathophysiology, we're going to see that again. Because we can study today filtration, okay, passive mechanisms or active mechanisms of transport, okay, as a concept, and give some examples. But then when we move to gastrointestinal, we are going to see how that works for the absorption, secretion. When we move to the kidney, in the process of urine formation, we are going to have filtration in the kidney, reabsorption in the tubules. Okay, some of these mechanisms are passive, some of them are active. Then we have the secretion. So we are going to be seeing that process in almost every system. Okay, so we have the homeostasis, relationship between structure and function at the cellular level, tissue, organ, etc. Then we have at the levels of organization, cell-to-cell communication. That's something that we are going to cover, not in too much detail. Okay, in pathophysiology, we are going to go deeper into that. how T cells talk to B cells, how macrophages and dendritic cells or other cells in the tissues interact, okay, how different organs talk to each other. Okay, then we have the genes to proteins, that is the flow of information. Okay, in physiology we study how things flow, how things move, matter, water, electrolytes, nutrients, how they move from one side to another. And there is also the flow of information. For example, from DNA to RNA to protein, or between different compartments. Okay, the importance of gradients, difference in concentration. Okay, most cases, for example, oxygen, CO2, sodium, potassium, they are going to move from high concentration to low concentration, following this gradient. Gradient means difference. Okay, and sometimes we have to move materials against the concentration gradient, so from low concentration to high concentration. And we have adaptation, okay, interdependence. There are many topics that we are not going to go too deeply, like, for example, the cell theory. Then we are going to go more deeply into cell membrane, how it works, what is the importance of the cell membrane. Okay, mass balance, that has to do with the inputs, outputs in different compartments. Okay, so if you want to maintain an equilibrium, balance, homeostasis, the inputs and the outputs must match. Okay, we can't have excess or deficiency of either or. Then energy, okay, different types of energy that we use, we store, we transform, okay, and how the body manages to maintain the energy balance. And then we have the topic of causality, which is a complex topic. And every time I see this causality, I think, okay, this is the part that is like in between the physiology and the philosophy. Okay, because to say that something causes another thing is like entering into another field. Okay, in most cases, you can point to a single cause. Okay, there are multiple factors that have to be present for something to occur. And then at the end we have the scientific reasoning. Okay, something that is one of our goals. Okay, is that you develop. Okay, I know many of you have it already. Okay, critical thinking skills, but you're going to develop even more. Okay, when you practice, if you want to practice medicine, as many other professions, you come to school to get knowledge, then you go to clinical rotations to develop skills plus that knowledge, and then when you start working and applying the skills and the knowledge, and with the interaction with other practitioners and patients, you will develop what we call the sense of authority. Okay, that is something that is at another level. What is that? That is that you are not going to be guided or moved by simple opinions of people. Okay, you are not going to follow TikTok guidance. I don't have anything against TikTok, but you know what I mean. When someone says, this is good for you, this is bad for you, and oh, let me change from the brand that I normally use. So every information that we receive is going to be analyzed, it's going to be weighted against evidence. Okay, it's not going to be simply accepted, and we're not going to give a publicity to any kind of information unless it is supported by evidence. And also, something that is even more difficult than that, we are not going to be afraid of being exposed to different opinions, different points of view. We are going to be thankful when someone tells us you are wrong. You made a mistake here. This is not accurate. Okay, I'm very happy for that. What is the guide me, enlighten me? Okay, according to new studies, this is better than that because of this. Oh, thank you for that. Okay, so remember, this is a profession, and you will never stop studying. Okay, and every day there are more than 500,000 new research articles published, and we can't read all of them. Okay, so we have to be open and, of course, judge. Okay, every new piece of information. Okay, before there was a Greek philosopher that said it is common among the educated minds, okay, to consider all the opinions without accepting any of them. Okay, that's the final goal of what we call the scientific reason. Okay, that you're going to be getting there little by little. So let's go back to the cells. Okay, we are going to be seeing the cell membrane. You know that is a double layer of lipids, phospholipids more exactly. It's described as being selectively permeable. Okay, determines what will enter, what will go out. That's a barrier that maintains separation between the intra and extracellular compartment, maintains the difference in composition and size, maintains that steady state. Okay, it's very important for homeostasis. It's in charge of transporting different nutrients, electrolytes, water. And we are going to be seeing the different types of transport. There is a diagram that we are going to see later that shows the difference between what we call the transcellular transport or the paracellular. And it's simply something, for example, when you have an endothelial layer, you want to move, let's say, glucose from one side to the other. Well, glucose has to pass one of the cell membranes, then pass the entire cytoplasm, and cross the other cell membrane to move to the other side of the epithelium. So it's through the cell. That is transcellular. Or glucose or other electrolytes or any other thing may pass in between two cells. Two epithelial cells may pass in between them. That is called paracellular transport. There is a diagram later. Now this transcellular transport, okay, will have different mechanisms. Some of these substances pass what we call passive transport, or actively. The difference is the requirement of energy. We're going to be seeing some examples. Diffusion is a type of passive transport. We move substances from high concentration to low concentration. And this is something that typically, for example, non-polar substances, substances without electric charge, tiny substances will pass readily through the membranes. They will pass through in between the lipids because they are lipophilic. Other substances, for example, large substances like large amino acids or electrolytes that have a charge, etc., will require a transporter. And when we move things against concentration gradient, we will need energy for that. It was frozen. Oh my God. Let's see. You have to stop there. Okay, here we have a diagram that shows the composition of the plasma membrane. Okay, there you can see the double layer of phospholipids. On the right side you have the image of how phospholipids look like, the diagram of course. They will have a hydrophilic head. What is that? The hydrophilic heads are looking at the outside and the inside of the cell membrane. They are in contact with water, hydrophilic. And then we have these tails, of fatty acids, hydrophobic tails. So the center, you imagine the cell membrane like a sandwich. On the center is going to be hydrophobic. So water can't be there. If you need water to pass through, there are specific channels for that. They're called aquaporins. And you see there are some other components. You see some proteins. Proteins can have some lipids or carbohydrates attached to them. So it can be divided into glycoproteins, okay, or lipoproteins if they have a lipid. Some of the proteins are just in the periphery, like floating. Okay, these are peripheral proteins. Notice that you can have some peripheral proteins in one side or another. They are totally different. Okay, we say that membrane is asymmetric. And then we have other proteins that are integral or transmembrane proteins. They span the entire membrane, go from one side to another. Okay, we are going to be studying some types of proteins. They can be channels, they can be transporters for different substances. In some cases, they transport one single thing, and we call them uniporters. They can transport two things. They can be symporters or antiporters. Okay, we have also cholesterol. Okay, the proportion between cholesterol and different types of fatty acids will give the membranes different types of flexibility. They shouldn't be too flexible. They shouldn't be too rigid. Okay, there has to be a composition, okay, that is compatible with all the cell functions, or all the cell membrane functions. And then we have the cytoskeleton. The cytoskeleton is something that when we study anatomy, physiology, we almost never cover in too much detail. Cytoskeleton is very important when we study some diseases. We are going to see it. Okay, there are different types of filaments that you can study, divided into intermediate filaments, microtubules, microfilaments. You have examples there. If you remember from the previous studies, actin, myosin, for example, these are types of filaments that belong to the cytoskeleton of the cells. We have an example here of intermediate filaments. Keratin, okay, protein that is present in nails, hair, the skin, okay, and also helps connecting one cell to another, forming a structure that we call desmosome, okay, and also provides strength, resilience, these special junctions between the epithelial cells. And they help the cells having their shape. They anchor the nucleus and organelles in place. Okay, well, the first time I studied biology and I studied all these organelles, I used to imagine the cytoplasm like a soup with all the organelles floating, and that you could shake the cell and they would go from one side, bouncing from one side to another. That doesn't happen. They are attached to different places. And the more you study that, the more you get amazed about how that works. For example, if you go to a muscle cell, okay, there are mitochondria. There are some mitochondria very close to the nucleus, other mitochondria very close to the contractile proteins, okay, but other mitochondria very close to the cell membrane, and they are different types of mitochondria with different functions, different capacity of making ATP. Okay, the cell membrane and the nucleus, they don't need the same amount of energy. Okay, in the cell membrane, you need to be providing for all these sodium-potassium pumps. In the nucleus, you don't need too much energy there. So this cytoskeleton is what maintains the mitochondria according to their type in one place or in another. Also, if you want to move something inside the cell, okay, these things will move using the cytoskeleton. Let's say, imagine a neuron. Okay, neurons can be very long. Some of them go from the cortex to the spinal cord, and then we have some that go from the spinal cord to the muscles of the legs. And neurotransmitters are made in the body of the neuron. Let's imagine someone who is like these basket players, okay, that extremely tall with those long legs. Okay, one of the neurons in the legs of these guys may be as long as 1 meter or 3 feet long. But the body of that neuron is in the spinal cord. Okay, and you have to send the neurotransmitters from there to be released there in the muscles. Every time someone is going to move, is going to walk, run. How you transport the neurotransmitters from the body 3 feet away, those neurotransmitters are going to be packed in some vesicles, and those vesicles have to travel okay, through along the axon until the end of the neuron. And they have to follow certain, like a highway. Okay, the microtubules in the neurons are gonna do that function. And if you see animations about how this works, you're gonna see these vesicles that have other proteins that transport the vesicles and look like feet walking on top of these microtubules. Then we have microfilaments, actin filaments. These are the thinnest one. Okay, they may exist in different forms. Remember the importance of actin for muscle contraction. They have a role in cell movement. For example, cilia or sperm cells, all of these cells that move, the neutrophils that have to migrate from the blood to places of infection, every cell that has to move, okay, is going to use these microfilaments, actin. And also these cytoskeletal proteins, something that is very useful from the clinical point of view, is that these, for example, the intermediate filaments that we mentioned, okay, they are going to be totally different depending on the tissue of origin. So sometimes we use the intermediate filaments to determine if someone has a tumor, for example, okay, and we suspect it's not a primary tumor of that organ, we can analyze these intermediate filaments to determine what is the tissue of origin. Yeah, I found a mass in the brain. This is not a brain tumor. Okay, let me look at the intermediate filaments. Oh, these are from the breast, or this is from the liver, this is from the colon. So that's another application of this. Here we have what I told you before, the feet. Okay, that or these motor proteins, we say feet because when you see the animations, they represent them like walking, okay, on top of these microtubules. Okay, these are motor proteins. They are different types. They are very large proteins that interact with the actin microfilaments. Okay, and there are two types, kinesins and dyneins. Okay, and the name is not as important as what is the function they do. Okay, they walk to transport things from the center of the cell towards the periphery. That is called anterograde transport. So let's say we are in the neuron, we are making acetylcholine, and we need to transport the acetylcholine to the axonal end. We are going to take those vesicles and give it to these kinesin proteins, so they are going to move it, the acetylcholine, to the axonal end. That acetylcholine is going to be released there, muscle contraction will occur, and then we are going to reuptake the choline part of acetylcholine, and we are going to reuse it. Sometimes we reuse it there to make more acetylcholine, and sometimes it is broken, it's damaged, and we have to repair it. So we're going to send it back to the nucleus. Recycle. Okay? We didn't invent recycling. That existed long before we thought about it. So we are going to use, in that case, the dyneins that transport proteins in the opposite direction. That's interesting. Okay? We also need to know that, for example, when someone gets an infection on the skin, herpes or rabies or tetanus, okay, some viruses that affect the nervous system, viruses will use these dynein proteins to go towards the brain, towards spinal cord, towards the brain. So it's not only how this physiologically helps in the physiological functions, is to know that also this can be used by viruses and or can be affected in different pathologic processes. So here we have pictures of how these things look like if we take cells and we stain the microtubules, microfilaments, and intermediate filaments, we can see how cells look like. Here we are looking at the cytoskeleton of the cells. And we are going to be studying in the future some diseases. Remember I told you many of these proteins help, okay, these epithelial cells to attach strongly to each other. Okay, they form tight junctions or desmosomes or hemidesmosomes or different junctions between epithelial cells. And we are going to see diseases in which we develop antibodies that instead of attacking viruses, bacteria, they attack these adhesion proteins that you see there. The main antibodies attacking that, so now the cells detach from each other. Okay, and people may have these potentially bad consequences. There are some diseases that have the same characteristics. They are called pemphigus vulgaris, or there is another that is bullous pemphigoid. It may happen in several autoimmune diseases. Another thing that is important, for example, when one of these cells in mind becomes a cancer cell, there are different mutations that may occur. There is a moment when these cells stop making adhesion proteins. And if cells don't have adhesion proteins, they are free to go and move to other organs. They may take the lymphatic vessels, blood vessels, okay, and that's one of the things that happens when people develop metastasis of cancers. And you may ask, well, if a cancer cell that is abnormal leaves a tissue and takes the blood, the lymphatic circulation, why that cell is not attacked by the immune system? Isn't the immune system responsible for detecting that? Yes, it is. But besides escaping, okay, from their origin, they also develop something that is very interesting. They take platelets and they surround themselves with platelets. Okay, they are like disguised. And when they travel, what the immune system cells see is platelets traveling. They're not going to attack the platelets.
A wolf in sheep's clothing. A wolf in sheep's clothing.
Exactly. They are disguised as a platelet.
When you
Would scalded skin syndrome be considered an impaired center, like an impairment of the cellular adhesions?
That occurs, yeah, but there are persons that don't have toxins, they're autoimmune. Okay, and skin scalded syndrome, there is more than that, okay, because of the action of the toxin and the inflammation there. In the diseases that I mentioned, antibodies specifically attack the junction protein. So it's like everything is intact, but you detach the cells. Okay? But yeah, there is also detachment, scalded skin syndrome. But we mentioned before the different mechanisms through which substances may pass across endothelial membranes or epithelial membranes. Then we have a diagram representing a generic epithelial membrane. Notice that the epithelial cells are very close to each other. There are tight junctions between them. There is an area that is looking outside. This can be the lumen of, for example, the duodenum, the gut. This can be the lumen of the bronchi. So it's the part that is looking at the outside. And then we have the basal or basolateral membrane, that is another name, that is resting on a basement membrane. Okay. Below that, we have the submucosa, or we have the dermis, depending on what tissue we are talking about. And you see that substances may enter, okay, through the cells or in between the cells. So we have this paracellular, para means to the side. Have the paralegal, someone who works with the lawyer. Paramedics, okay, someone who works together with us like parallel. And then we have the transcellular. Notice that when you study the transcellular transport, there are two membranes that have to be passed. Okay? The epithelial and then the basolateral. So that substance has to move through two cell membranes. And there are different mechanisms for that. And we have here the most important mechanisms. Okay, some substances will move through the lipid bilayer, free oxygen, CO2, carbon monoxide, nitric oxide, substances that are non-polar, small gases, for example. other substances will move through transport proteins. Okay, for that we have a transport mechanism that we call facilitated diffusion. Substances that have a charge, for example, or large substances like glucose will need a facilitated diffusion. Osmosis is another example. Water will move using channels that we call aquaporins. In some cells, the aquaporins are always there. In some cells, aquaporins are not there. We have to place them if it's necessary. So the body adapts to the condition, so we can either absorb water or not, depending on if we place or we don't place aquaporins in the cells. Now, all these things that we have mentioned are mechanisms of transport that we call passive. No energy is needed for those. Substances are going to move from high concentration to low concentration. In the case of osmosis, the driving force is the osmotic pressure. Okay, water will follow the compartment that has the greatest osmotic pressure. That means salt or sugar. Okay, when we talk about medicine, if we were studying chemistry, if we were studying how to do things in a laboratory or at the industrial level, we would be talking about other types of osmosis. But for us, osmotic pressure means salt or sugar. That's it. And there can be more things, but normally. Now, mechanisms of transport that use ATP, or active transport. Active transport is divided into two types, primary or secondary. Okay, primary active transport is when you have a transporter that uses energy to move a substance against the concentration gradient. For example, sodium-potassium pump is the classic example. It's going to take sodium out of the cell against the concentration gradient and potassium into the cell against the concentration gradient. We have more calcium pumps, okay, the hydrogen pump in the stomach that we call the proton pump, that is pumping hydrogen into the lumen of the stomach against concentration. Is the one that we inhibit when we take omeprazole, proton pump inhibitor. Okay, so that's what we do. Now, we're going to be seeing some examples of secondary active transport. Okay, and that means it's very understood when we see the diagram to see what it means, primary or secondary, because they have to be acting together. Okay, but the most important thing is the secondary active transporters don't directly use energy. They don't use ATP, but they use a gradient that is generated by the active transporters, by the primary active transporter. Okay, we're going to see that in the diagram. And then we have the transport within vesicles, membrane vesicles, that can be endocytosis or exocytosis, okay, that are mechanisms of transport that use ATP, okay, so they are considered energy dependent. And here we have a representation of the mechanism of simple diffusion. I don't know if we have represented that properly, but these are like 3 steps in time. What is that? We are going to follow time. Okay, when we start, we have a great concentration of a substance, any substance, oxygen, CO2, on one side of the membrane. So the rate of transport is very big, it's large, because the difference in concentration is huge at the beginning. This applies to our membrane, but this could apply to any membrane that is permeable or freely permeable to this substance. Then as time goes by, we are going to have more concentration inside than at the beginning and less outside, so the gradient is not so, the difference is not so big. The rate of transport is decreasing. You see the arrow smaller. And at the end, we see the exact same concentration of substances at both sides of the membrane. So now you have a double arrow, meaning there is no net transport. Something goes out, that will go back. Okay? It's not that. This doesn't mean that there is no transport. There is no net movement. Okay? It's always maintaining the equilibrium. Substances can move freely, but always everything is gonna go back to the equilibrium. Now here we have a representation of other types of passive transport mechanisms. Again, we have the simple diffusion, exactly the one we saw before, okay, through the cell membrane. And then we have a couple of mechanisms, a channel that will allow certain specific things to pass. Okay, some channels are specific for sodium, others are specific for chloride, others will be specific for potassium, calcium, etc. They are very selective. There is only one exception, and we're going to study that in cardiovascular. There are channels that allow both sodium and potassium to move through. And they are exceptional, so exceptional that we call them funny channels. The people who discovered them, that's the name they gave to these channels, funny channels. A channel that allows two ions to move through. It's not that specific. And then we have another on the right, it's a transporter that we call a uniporter. Notice that it's not a channel, it's not open, okay, it's not totally open like a channel. Okay, it's a transporter that when a substance binds to it, it will change the configuration, and the substance is gonna like rotate and enter inside the cells. Once the substance is released on the other side, it will take the original configuration. Now, we have to mention one specific type of mechanism that allows the transport across membranes, and it's the filtration. Okay, something that we are going to study more specifically when we move to the kidney. Filtration is a process of movement of water and several other substances. For example, when we filtrate the blood in the glomerulus, okay, almost everything that is in that blood will move to the Bowman's capsule. Water, sugar, sodium, creatinine, urea, uric acid, amino acids, vitamins, everything is going to move. Everything that fits in between the little holes of the epithelium will move to the Bowman's capsule. This type of transport in which we move water and everything else is called bulk transport. Bulk transport. And that filtration, okay, will occur following a gradient. There is concentration gradient, etc. But the most important gradient here is a pressure gradient. The hydrostatic pressure that is created, we are going to study in the future, by the difference in diameter between the afferent and efferent arterioles. Okay, glomerulus, you have the efferent arteriole. Okay, the efferent is the exit. If the exit has a smaller diameter than the entrance, the pressure in that loop of capillaries is going to be huge. It's like when you're making espresso coffee. You need to create pressure to filtrate the coffee. If there is no pressure, there is no coffee. All the vapor will escape, and so the pressure there is very important. Because notice that for that filtration to occur, okay, there has to be a predominant pressure that allows or favors filtration, and that is the blood hydrostatic pressure. Okay, there are forces that are opposing filtration, and one of them is the blood colloid osmotic pressure, also known as oncotic pressure of the blood, that is given by the amount of albumin that we have in the blood. Albumin is a protein that keeps the water inside the blood vessels. Okay, so the blood pressure, hydrostatic pressure, is pushing the water out, but the albumin is keeping the water in. So there has to be a predominant hydrostatic pressure okay, for the filtration to occur. There is one example. In this case, the blood hydrostatic pressure is 55 millimeters of mercury, and we have two opposing pressures. One is the oncotic pressure, and in this case, we write there 30 millimeters of mercury, and also we have to consider the hydrostatic pressure on the other side of the membrane. Bowman's capsule hydrostatic pressure, we are giving a value of 15. Okay, when you add up all these, okay, you have a net filtration pressure of 10 millimeters of mercury. Why is that important? Because if something, for example, decreases, let's say someone has a hypotension, and we decrease the blood hydrostatic pressure from 55 to 45. Now, what is the filtration pressure? 45, 45, no filtration pressure, zero. That's why when people are extremely dehydrated, hemorrhage, shock, they don't produce urine, because there is no pressure to filtrate the blood. And that's why the priority when someone goes to the ER in shock is fluid, fluid, fluid. I don't care why is the person in shock. Fluid, 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 epinephrine, cortisol, until we save the kidneys. Then we can take care of everything else. Okay? And also, the hydrostatic pressure on this side is very low, it's 15, because the urine is flowing, proximal tubule, loop of Henle, distal tubule, bladder. But what happens if I place an obstruction here? That pressure here, the kidney makes urine. Goes from 15, 20, 30, 35, until the two pressures are equal, and we don't make any more urine. Okay, so that's the importance of this. We're going to be seeing examples when we get to the kidneys, and also in pathophysiology more than in physiology. Now, diffusion is affected by several factors. Okay, and this is something that you have seen for sure. We already mentioned the concentration gradient. As you saw in the diagram, the greater the gradient, remember gradient is difference. The greater the rate of diffusion. Okay, the greater the difference, the greater the diffusion rate. Then we have the mass of the molecules, heavier molecules go more slowly, something that makes sense. Okay, the temperature, greater the temperature, faster the rate of diffusion. The density of the solvent, the density increases, everything moves more slowly. And for that, you always try to use analogies from normal life. You have regular milk and condensed milk. Which one flows faster? That's more or less how we, and if you have condensed milk, it's in the refrigerator, it's very cold. Will it go faster or more slowly?
Slower.
Now you have the temperature, the density, okay? Now, for us in medicine, the most important things to consider are the first one, concentration gradient, and the last thing, surface area, thickness of the membrane, and distance that the substances have to travel. We are not working in the industry creating dyes or colorants or anything that we are not creating detergents. We don't have to work with thousands of chemicals at different temperatures, concentrations, different purposes, colors. No, we are working more or less constant system. Okay, that almost everything that you see here will stay the same. Temperature, our temperature increases, decreases, but not that much. So some couple of degrees above, below. Now, we are going to be seeing examples of how the surface area, for example, healthy person, the surface area of the lung, or of the alveoli, if you put them together as a flat surface, Okay, in every book you hear the same about everything. It's like 5 football fields, or like 8. It's a lot. Okay, now if someone has emphysema, emphysema is characterized by destruction of the alveoli. That surface may be reduced by half, okay, or a third. Of course, there's going to be more diffusion the greater the surface. The same with the intestine. Small intestine is huge, and with all of these villi, microvilli is even larger. If someone has, we have to remove part of the small intestine. The surface for absorption is a lot smaller. The thickness of the membrane. For this, we are going to consider the alveoli. The alveoli are a very, very, very thin layer of epithelium. Okay, squamous epithelium. Now if you start adding there, let's say, mucus, or start adding there pus, or inflammatory cells, okay, the thickness is going to increase. Or if someone develops fibrosis of these, of the lungs, we're going to increase the thickness. So the diffusion is going to be slower. And then we have the distance that the substances have to move through. The greater the distance, the slower the traffic, the slower the speed. And for example, when someone has edema, that is going to become very important, because edema means presence of water, excess water in the interstitial space. So now the cells are going to be farther away. So if you want to move any chemical, any signal between cells, maybe they don't reach the other cell. Okay, if oxygen has to move, and maybe some cells are now a lot farther away than if there wasn't edema. There are some tests, of course we don't do in the daily practice, but there are tests in which people measure something that we call the intercapillary distance. Okay, this is done in certain research. This is not done in the clinical practice unless you need it for a specific reason. I'm representing here a couple of blood vessels. There is a cell. That cell can easily get oxygen from both of these capillaries. It's happy receiving oxygen from these 2 sources. On the other side, okay, we see that there is a greater intercapillary distance, and that may be due to the presence of excess water, in the case of edema. Okay, now we have a very important, very huge intercapillary distance. Now maybe the oxygen doesn't reach that cell, and also there is no possibility of eliminating the toxins that that cell is producing. So the cell produces waste products that stay there, and it's not getting the oxygen. Of course, if this is maintained, okay, for a long time, that cell is absolutely gonna die. And here we have the differences, okay, in the kinetics of simple diffusion versus facilitated diffusion. Okay, I'm sure you all more or less, you look carefully, you're not going to see the simple diffusion because it's down there, like a flat line, okay, that increases a little bit. Okay, it's important in physiology and in everything that has to do with science, learn how to read graphics. Okay, in this graphic, for example, in the y-axis, okay, we have the rate of transport that increases as we move up. And on the x-axis, we have the external solute concentration. Okay, so we are adding more solute, let's say salt or sugar or anything that you add to a solution. Notice that simple diffusion is going to increase in a linear way as we increase the concentration of the solute, and that is how much it increases compared to the facilitated diffusion, to the movement of substances when we use a transporter, channel or uniporter. Okay, so it's very efficient, the transport of substances via uniporters or channels. The problem is that these transporters are saturated. We have a limited amount, okay, in the cell membranes. Just to say a number, let's remind that a cell, X cell, has 20 transporters for glucose. And these transporters are capable of moving glucose 1 molecule per second. Okay, so each transporter is going to move 1 molecule of glucose per second. Okay, and we start adding glucose. That's going to occur very fast, so that the transport of glucose is going to increase very rapidly. But when the 20 transporters are transporting glucose, you have to wait until they are free again to transport 20 molecules more. Doesn't matter that you add a kilogram of glucose. The transporters have their pace. It's like when you come back from any travel, okay? You're there waiting in the immigration line. Doesn't matter that there are 20 people or 20,000 people. They are gonna go one by one, look at the passport, look at the fingerprints, make a picture, next one. Doesn't matter how many people are there. Let's take a look at this before going to the break, okay? Here we have osmosis. Osmosis is simply the movement of water following a gradient, but in this case the gradient is osmotic pressure gradient difference. Okay, we have here the example that you have seen in many books for sure. Okay, that we have a membrane that is only permeable to water. That membrane cannot be permeable to the solutes, because if the membrane is permeable to the solutes and water, this is not going to happen. Notice that we have here 4, and here we have many. Okay, and the water is going to move from following the gradient, following the osmotic pressure gradient, to the place with greater osmotic pressure. And the same thing is going to happen in our cell membranes. Our cell membranes are permeable to only non-polar small molecules. Okay, they are not permeable to sodium or potassium. And in the case of glucose, okay, glucose is a substance that normally when we eat or drink something that contains glucose, glucose is going to be taken immediately into the cells. In many cells, glucose is going to enter without help of anything. But some cells, skeletal muscle and adipose tissue cells, won't take glucose inside unless insulin is present. So in the case of a healthy person, when we eat something that contains sugar, that sugar is going to go into our cells and it's not going to affect our blood osmolarity at all. But if someone has diabetes mellitus, that glucose is going to stay for a very long time in the blood, and it's going to increase very importantly, okay, creating an increased osmolarity in the blood. Now, our blood osmolarity can change because of excess glucose or because we are losing free water. Okay, we have a hormone that is antidiuretic hormone that typically keeps the water inside the body. It tells the kidneys to retain water. If that antidiuretic hormone is not present or is not working, our kidneys are going to eliminate water, free water, and the sodium is going to stay inside the body, making our sodium very high. That also increases the osmolarity of the blood. And when we have a very high osmolarity, our cells are going to start suffering. Okay? Cells are going to start losing water. For example, the red blood cells. Okay, the water from the inside of the red blood cells is going to move out. They are going to start shrinking, and we may have hemolysis. Same thing is going to happen in the neurons. Neurons start shrinking, and that's not good. Because if the neurons start getting reduced inside, let's imagine these are a couple of neurons in the brain, they are typically in contact. And if they start shrinking, what happens? They don't talk anymore. And that's why we are going to have certain symptoms like confusion, different types of severity, and even coma as a result of these neurons not communicating with each other. Okay, it's 2:30, and we can have a break until 2:45. Okay. Okay. Let's talk about something. Let's move to the second part now. And here we have a concept that belongs to both the passive-active transport mechanisms. I didn't know where to put that slide, so I put it in between. And it's a concept, or it's a piece of information about a specific type of channel, ion channels. We're going to be talking, starting now, about ion channels. Remember, they are specific for certain ions, with the exception of the phonic channels, that we're going to talk about them in the future. And there are different types of ion channels. In most cases, they are simply like a hole in the membrane, okay, made of proteins, that only certain ions will fit according to their size, charge, etc. If you remember from the periodic table, the atomic radius, ionic radius, all these things apply here in the selectivity of these channels. They have different sizes. We have to talk, before moving to that topic, about the mechanisms that allow the opening, closing of some of these channels. Okay. We're going to see that there are channels that are always open. We call them leak channels. Okay, if you take a cell membrane, let's imagine we are in a neuron. Okay, we are in the dendrite of a neuron. We are going to see several sodium and potassium channels that are always open. Okay, so sodium is going into the neuron and potassium is going out of the neuron, and the sodium-potassium pump has to be taken. Okay, sodium, go away, potassium, come back. It's all the time taking everything to their compartment. But there's always a leak of sodium and potassium. More potassium than sodium. Let's say, just inventing numbers, there are twice as many potassium channels than sodium channels. What does that mean? That there are going to be more positive ions going out than positive ions coming into the cells. And that creates a more negative environment inside the cells, because you have more potassium moving out than sodium entering into the cell. But those are always open, leak channels. Okay, that's what that phenomenon, together with the sodium-potassium pump activity, is what creates what we call the resting membrane potential. It's different for different cells. And then there are channels that are closed, are gated. They have gating mechanisms. Okay, that only open if we stimulate them either with a chemical or with a change in the voltage of the membrane, or by stretching the membrane. Okay, so there are ligand-gated channels. For example, if you bind acetylcholine to them, they are going to open. When acetylcholine is removed, they close. Or if the voltage of the membrane goes from -70 to -55, they open. And when the voltage goes to +10, they close. Okay, depending on the voltage, they are going to open or close. So we have ligand-gated channels, voltage-gated channels, and some of them open when you stretch. You remember, for example, the cilia in the inner ear, or in the labyrinth. Cilia move, bend in one direction or another. Or when we have a wave, a sound wave, you have this basilar membrane, okay, vibrating or moving inside the inner ear. That is going to produce the bending of the cilia of the cell. That bending opens channels and closes channels. Opens and closes channels. When we stretch the blood vessel, the endothelium, that stretching will open channels, and then they will close when it goes back to normal. Those are mechanically gated channels or stretch-activated channels. Now, we have special channels. That's why we have this image here. Notice that they have several mechanisms. They have a double gating mechanism. They have a selectivity filter, okay, so only sodium will pass. Potassium can't pass, chloride can't pass, calcium can't pass, only sodium. Okay, but that sodium will pass, okay, only if the two gating mechanisms are open, double gating for sodium channels. Okay, so we are going to see when they open, when they close, okay, and what is the importance of that double gating, okay, when we study more in detail the action potential of the neurons and also the action potentials in the contractile cells in the heart. I just keep that information in mind. Now, we can move to the active transport mechanisms. Okay, when we move substances against their concentration gradient. Okay, and we are going to be seeing here what are the active transport and secondary active transport mechanisms. The primary active transport is, it's not hard to understand. It's just taking the same mechanism that we saw for the facilitated diffusion, but now moving the ions against their concentration. And for that we need ATP, okay, to move something against their concentration gradient. Now, the transporters that we classify as secondary active transporters don't directly use ATP. Okay, they use the gradient, okay, that is created by this primary active transporter, okay, to move a solute in the direction, okay, of its electrochemical gradient. And this always involves carrier proteins, can be uniporters, symporters, or antiporters. So there are different examples of this that we have here. On top here, we have examples of primary active transporters. These transporters are also called ATPases because they have enzymes that break down ATP, so make ADP with the ATP. Okay, on the left, we have the sodium-potassium pump. That is moving 3 sodiums out and 2 potassiums into the cell. Okay, different directions. Then we have a second one, is hydrogen-potassium ATPase, is pumping hydrogen. That's the example that we mentioned, the proton pump. Just remind that on that side, we have the gastric acid. The pH in this part is 1 or 2. That means there is a very high concentration of hydrogen. So you have to move hydrogen against the concentration gradient in exchange for potassium. Okay, notice that we have to put a cation, a positive charge out, and take a positive charge, okay, to maintain the electron neutrality of the cells. Then we have a calcium pump. Calcium pumps are important in the smooth muscle, skeletal muscle, in the sarcoplasmic reticulum. They are called calcium ATPases. There are some of them present in the sarcoplasmic reticulum to take the calcium back into the sarcoplasmic reticulum. Okay, and then we have other transporters for organic solutes. I'm not going to enter into that now. I'm going to give it in the basics. With the B in the center, we have examples of secondary active transport. Okay? Secondary active transport. They are transporting solutes in the same direction. Okay, and we have exchangers, okay, that transport solutes in opposite directions, also known as antiporters. Okay, when they transport in the same direction, you can call them symporters. Okay, in different directions, antiporters. We have here the example of smooth endoplasmic reticulum. And that we mentioned before, the calcium active transport in the smooth endoplasmic reticulum. And this is the proton pump that is pushing or pumping hydrogen ions into the lysosomes. Lysosomes also have a very acidic environment. Remember, this is the digestive organ of the cells where we break down all the debris and bacteria. And below here, we have different types of syn-porters on the left and anti-porters or exchangers on the right. Notice that these proteins in blue, they are not using ATP as the ones above. Okay, they are moving, for example, here we have the sodium glucose cotransporter. Okay, we are going to study that in pharmacology. Sodium glucose cotransporters that, for example, serve in the kidney to reabsorb glucose. We filtrate the urine, that urine contains lots of glucose and sodium and many things. If we want to take the glucose back into the blood, we cannot take the glucose alone. We have to take sodium with it. Okay, so we are going to move sodium and glucose together. Now, how we do that? Well, the cells that are taking the glucose and the sodium, they need to be almost empty of sodium. There can be sodium inside the cells. And that lack of sodium inside the cell is created by the sodium-potassium pump. The sodium-potassium pump is taking the sodium out of the cells, creating a very low concentration of sodium here. So we can take the sodium and the glucose, okay, through this syn-porter. The same thing happens in the intestines when we absorb glucose. It has to be combined with sodium, this co-transport, for the absorption of amino acids too. We eat a steak, we break down all the meat, all the proteins into amino acids. To absorb those amino acids, we need to combine them, okay, with glucose, no, with sodium, sorry. Sodium amino acid cotransporters. There are more sodium chloride cotransporters, sodium peptide or amino acid cotransporters for the absorption. And on the right side, we have exchangers, okay, anti-porters. We can exchange sodium for hydrogen. And this is, for example, you have studied, I'm sure in the past, well, the kidneys eliminate excess hydrogen, okay, to regulate the pH of the blood. If the blood is too acidic, they eliminate hydrogen. Well, look what it does. Hydrogen, yes, in exchange for sodium. So we reabsorb sodium and eliminate hydrogen. And you have there other examples, sodium, calcium exchanger, bicarbonate, chloride exchanger. We're going to see the implications of this when we study certain acid-base disorders. Okay, let's say someone has diarrhea. When we have diarrhea, we are losing lots of bicarbonate. We lose bicarbonate, that is an anion. Our body wants to replenish the anions that we are losing. So what the body does is starts reabsorbing chloride. Okay, you're going to be understanding this when we study the different diseases in the future. Okay, and that way, that's something that we can use to differentiate when the person develops acidosis, if it's because of diarrhea or because of other things. Just by measuring the chloride in the blood. You're going to see there is an metabolic acidosis that is called hyperchloremic. Okay, so that has applications. Those physiological phenomena, okay, will have applications in the future. And this is representing simply how the sodium-potassium works. Okay, producing a change in the configuration of the sodium-potassium ATPase. If you follow from left to right, okay, you are going to see the sodium-potassium pump. It's a protein, okay, that is open to the inside of the cell. It grabs 3 sodium, okay, and then turns around, grabs an ATP. ATP is broken down, okay, for it to take the potassium and then turns around to release the potassium inside the cells. That's constantly working. Okay, if you go to In YouTube, I think there are videos that show that, and even they show videos taken with electronic microscopy that you can see the actual sodium potassium pump working. It's like an engine rotating all the time using 1 ATP per cycle. That's incredible. And here you have an illustration of the mechanism of secondary active transport. Okay, we have on top the top part of the membrane. Let's imagine that this is a cell in the small intestine. Okay, we ate something that contains glucose, contains sodium. Okay, and notice that glucose is the green thing, and a couple of sodium are gonna fit there in that transporter, that sodium glucose C-importer. Now, the sodium glucose C-importer is not gonna work unless it has the glucose and the sodium, and the concentration of sodium inside the cells is very low. So we have here the sodium potassium pump taking sodium away, so we can so the low concentration of sodium drives the uptake of glucose, or the absorption of glucose in this case in the small intestine cells. Then when glucose is inside the cells and has a very high concentration, there are other transporters on the other side of the membrane that will take the glucose to the blood. Something that we are going to be studying in the due time. There are a type of transporter on the apical part of the cell and another transporter in the basal part of the cells.
So the sodium potassium pump would be on the base of the
basolateral membrane. Yeah. Okay, concept. It's importer and importer. And of course we have to mention other mechanisms of transport. Phagocytosis, endocytosis, exocytosis. Okay, these things are concepts that we are going to explore more in detail when we move to immunology. Okay, see how macrophages or dendritic cells or any phagocytic cell is capable of recognizing microbes. Okay, our phagocytic cells have receptors that we call toll-like receptors. The first time I saw that name, I mined the toll in the expressway, but it has nothing to do with that. And I mind that it was because of that, because these receptors recognize a pattern, like a barcode. Okay, they are looking for certain patterns that are present only in bacteria, viruses, etc. For example, peptidoglycan, double-stranded RNA, things that we don't have. And when they detect that, they are activated. And I thought, it's like a toll. They see car or with the device or some pass, it's activated. I said, oh my goodness. But no, it has nothing to do with the toll. Well, these toll-like receptors are going to bind to bacteria or anything that is abnormal. Okay, the affinity of these macrophages for these microbes is going to be even greater if the pathogens are surrounded by antibodies. Okay, the macrophage is going to grab it more strongly. And there you see how once the binding of the microbe to the toll-like receptor occurs, okay, we have this macrophage starts making like evaginations of the membrane and engulfing the bacteria, making what we call a phagosome. Then the phagosome is going to join with another organelle that is the lysosome, forming a phagolysosome. Okay, bacteria is going to be digested by these enzymes inside the lysosome. At the same time, these macrophages are going to start producing different inflammatory cytokines, telling everybody else that they are eating the bacteria, attracting cells, attracting different things, activating the immune response. And then we have the processes of endocytosis and exocytosis. Okay, that is simply taking things inside the cells or eliminating things. Okay, the endocytosis then is subdivided into pinocytosis, phagocytosis. We are not going to right now enter into those distinctions. I told you, in some aspects, the clinical physiology is easier than the one you had before, but in some other aspects, it's not as easy. It goes more in detail into other stuff. Here we have a cell that is like taking material from the exterior, forming this vacuole, this vesicle, okay, like the one we saw before with the macrophage. Okay, and on the right side, you have a cell that is eliminating things. This can be waste products, or this can be simply signals, for example, neurotransmitters. When we have a neuron that releases neurotransmitters to produce the muscle contraction, that is exocytosis. Okay? This communication between cells occurs after the release of different things. Okay, these substances may act nearby, and what we call how's that? Neighboring cells? I forgot. Huh? Paracrine. Or at a distance, that is endocrine. Okay, thank you so much for the support. And if you see in the notes, and let me tell you something. In some slides, you're going to see that I give information in the notes down there. Nothing that is in the notes will be in a test. Okay, those are things that are in advance for pathophysiology or better understanding. You don't study using the notes. Okay, and for example, right now we are sharing the PowerPoint using the drive, but in Canvas, okay, if the PowerPoint is too large, we can't share the PowerPoint. We have to share PDF. If we convert the PowerPoint to PDF, there is no notes, there is nothing. Okay, so only the things that appear on. I mean, because there I'm seeing that it's talking about Lambert-Eaton syndrome, it's talking, that's not going to be in any test. You can read it, you can, if you have time, you can look for information, but you don't study Lambert-Eaton. I remember when I was teaching in a nursing school, was the very basic gen-ed anatomy physiology. The lecture was red blood cells are for transport of oxygen and CO2, and white blood cells defense, platelets, hemostasis, and then the white blood cells are subdivided, leukocytes, neutrophils, lymphocytes. And I started mentioning, for example, when people have leukocytic leukemia or lymphocytic leukemias, it's because this type of leukocyte or this type of lymphocyte, etc., acute, chronic. And the next week there was a quiz, a very simple quiz. Which of the following cells transports oxygen? Which of the following cells performs a plug when there is a wound? And one student that was a very good student, she failed. She failed. What happened to you? Oh, professor, I spent the entire night studying leukemia and lymphoma. Did you read the learning objectives? Did you see leukemia and lymphoma there? Oh, professor, but that was so interesting about them. You learned your lesson. And the same, well, you don't have too many resources here. Here what I do is I put some videos, some stuff there. Have you seen this video before? Have you seen this video? That is a macrophage chasing a bacteria. phagocytosis.
Now we can just slide it to the other
No?
No. Because our
That's good. That's good. Well, you can watch it later. Internet is helping us.
Professor, if you click Control Print for the PowerPoint and you click on Layout, you can include the notes.
In the PDF,
Yeah.
can you send me an email? Because for sure I will forget in 30
I'll
seconds.
send you an email.
Please send me an email. Control print. Control print.
Control Print and then Layout, and then it'll show a drop-down thing, and you're gonna put notes.
Like when you're printing and it shows you a preview.
Okay, thank you.
The only thing is it'll make the slide
Oh,
a little hard.
you can't upload it like that.
So, you want Control Print? I know. So where do you put the notes? Oh no. Oh, there we go. And you're gonna put Layout, and you're gonna put in the notes. Actually, right on my face.
And we have this light too.
Yeah, I'm not loading.
So then the slide makes it small.
When you zoom in, it's perfectly fine. So, like here, I have an idea. I'm looking at it. I'm gonna bring my computer in. Yeah, it just happened right now. Right now. I've been having to type them
So it's a PDF.
Yeah, I think she would have been and the quality. So you want to drive or I don't know.